Hi, this is our experiment about sun-adapted and shade-adapted plants. I'm Lola.
I'm Summer.
And I'm Maya. Our experiment is focused on whether or not our plant, the tall evening primrose, is sun-adapted or shade-adapted.
First, we'll discuss the black sage, which is a sun-adapted plant, also known as the Salvia mellifera. It is a small shrub in the mint family, and it grows light purple flowers. It grows on the south-facing slopes of the Tecolote Canyon, which receive a lot more light than the north-facing slopes. It grows best in full sun and is drought tolerant, according to Kent in the 2022 findings. Like many other sun-adapted plants, these leaves are small and thick, and they contain high levels of a photosynthetic enzyme, which is responsible for a high rate of photosynthesis in sun-adapted plants, and also a high rate of carbon dioxide fixation, which is where the plant converts light energy to chemical energy.
The lemonade berry plant is a shrub that is shade-adapted and commonly grows on the north-facing slope of the Tecolote Canyon. The lemonade berry has oval-shaped, thicker dark green leaves with a smooth texture and pinkish-purplish veins. The plant also produces berries with a bright reddish-orange color that give off a lemon citrus taste. Lemonade berry plants grow best in partial sun and low moisture.
The black sage plant commonly grows on the south-facing slope of the Tecolote Canyon. Meanwhile, the lemonade berry grows on the north-facing slope. These canyons receive more direct sunlight than the north-facing slopes, especially in the winter and early spring when it rains.
When comparing shade and sun-adapted plants based on previous research, shade-adapted plants have higher chlorophyll concentration than sun-adapted plants. Chlorophyll helps plants absorb sunlight, so shade-adapted plants need more to absorb maximum energy from the sun from the little sunlight they receive. Shade-adapted plants have less robusco concentration, the enzyme that starts the process of turning light energy to chemical energy, than sun-adapted plants. Shade-adapted plants have lower rates of enzyme activity and photosynthesis since they have less photosynthetic enzymes than sun-adapted plants. Shade-adapted plants are known to have a higher stromatal density as compared to sun-adapted plants, which makes them more efficient at CO2, water, and oxygen exchange.
The primrose plant is often tall with a central stem with alternate leaves. The stems are pale green or crimson in color and have white hairs on them. The light or olive green leaves can grow up to 8 inches long and 2 inches wide, but they are usually smaller. The leaf margins are smooth or slightly dentate, and the leaves are virtually hairless. The plant's apex bears a panicle of pale yellow blooms.
Based on our research, we think the unknown species, the tall evening primrose, is sun-adapted because according to the internet, it is best grown in full sun conditions. This plant also prefers well-drained soil and can survive in dry conditions. These characteristics are similar to the black sage plant, which is sun-adapted.
These questions are important as they can help scientists understand where plants grow best. Because of global warming, loss of habitat and native species, and an unbalanced ecosystem, it's extremely important that we help plants flourish by giving them their optimal conditions. Understanding the different mechanisms of shade-adapted and sun-adapted plants can help biologists understand why and where certain plants can grow the best. S. Mothler et al. Explore similar methods and ideologies in their publication Photosynthetic Efficiency in Sun and Shade-Adapted Plants.
Based on leaf morphology and analysis of other sources, we came upon 3 hypotheses. Hypothesis 1 states that since the black sage is sun-adapted, it will have significant traits linked to sun-adapted plants. Hypothesis 2 states that since the lemonade berry is shade-adapted, it will have its own characteristics linked to shade-adapted plants. Based off of hypothesis 1 and 2 and observations of the tall evening primrose, we hypothesize that the tall evening primrose is sun-adapted.
4 parameters were measured. These were chlorophyll concentration, which was measured using a chlorophyll meter, protein concentration, which was measured using a blue assay on the leaf discs, and the photosynthetic rate using the erga machine. Lastly, the stomata count was measured using a microscope and observing the stomata in there.
Methods and materials. To record chlorophyll concentration, we used a chlorophyll meter on a sample size of 4 leaves to create an average chlorophyll concentration of each species. Next, to record the robusco protein concentration, we made a Bradford assay and standard curve for the absorbance and concentration values of 6 standard dilutions using the spectrophotometer. The standard curve was then used to measure the Robusco concentration in a sample size of 6 leaves from each plant species. To measure the photosynthetic rate, we used the ergo machine on a sample size of 3 leaves from each plant at 6 different light intensities from full sunlight to full darkness. The data from this technique was graphed into a light response curve and a quantum efficiency graph. The stomata count for each plant was measured at a sample size of 10 points on each of the plant species leaves. This was done by applying nail polish to the underside of the leaves and removing it when dry, which peeled off a layer of the epidermis, which showed stomata when placed under a microscope.
Averages of chlorophyll concentration of the lemonade berry, black sage, and tall evening primrose are shown. Lemonade berry plants showed the highest level of chlorophyll concentration compared to the black sage and tall evening primrose. Both lemonade berry and black sage had no significant difference in chlorophyll concentration. Primrose had a significantly lower concentration than lemonade berry and black sage plants. ANOVA tests show evening primrose had a significantly lower concentration with a p-value less than 0.01.
The photosynthetic rate was measured using a light intensity curve comparing light intensity versus photosynthetic rate between the 3 plants. The highest photosynthetic rate was black sage with a 21 umol CO2 M2S1. The second one was primrose having a 16 umol, and then lemonberry having 2.5 umol. The quantum efficiency, the highest was the black sage, having a quantum efficiency of 0.059, the primrose having a quantum efficiency of 0.0129, and the lemonberry having a quantum efficiency of 0.0246.
The averages of the Robusco protein concentration in each of the 3 species is shown by the graph above. As you can see, the lemonade berry showed the highest Robusco concentration of about 0.0444 milligrams. The black sage had the second highest Robusco concentration of about 0.0421 milligrams. However, there was no statistically significant difference between the lemonberry and the black sage concentration levels. The tall evening primrose had a significantly lower Robusco concentration than the lemonade berry and the black sage. The concentration of the tall evening primrose was about 0.0110 milligrams, and this difference was statistically highly significant to both the lemonade berry and the black sage.
The graph shows comparison of the number of stomata found on each leaf sample of the lemonade berry, black sage, and primrose plants. The lemonade berry plant was shown to have the highest number of stomata observed as compared to the black sage, with a p-value less than 0.01. The tall evening primrose with a p-value of 0.01 as well. The black sage had a higher stomata count observed as compared to the tall evening primrose, with a p-value less than 0.05.
For hypothesis number 1, black sage, we predicted it was sun-adapted. This hypothesis was accepted as it had a high photosynthetic rate and high protein concentration. On the other hand, it had lower stomata and low chlorophyll. We hypothesized the stomata differently. We thought it would be higher, but this could have been just a calculating error counting under the microscope. For hypothesis 2, lemonade berry, we hypothesized it was shade adapted. This hypothesis was accepted, which had a high chlorophyll concentration, high stomata count, and high protein concentration. Rather, it had a low photosynthetic rate. And for hypothesis 3, we hypothesized the tall evening primrose was sun-adapted, which was partially rejected as it had a high photosynthetic rate and low chlorophyll concentration, stomata count, and protein concentration.
Our results show that the lemonade berry, which is a shade-adapted plant, had higher chlorophyll concentration than the sun-adapted plant, which is in agreement with what we previously knew. It also shows that the lemonade berry, which is a shade-adapted plant, had a higher Robusco concentration, which did not agree with what we previously knew from previous research. Recall, however, that the data between the differences of the lemonade berry and the black sage were not significant. In addition, moderate heat stress can cause deactivation of Robusco, so it's possible that the light-adapted plants have decreased Robusco. However, since 2 plants are relatively in the same environment, the temperature difference would not be drastic, thus their difference is not significant. The black sage had the highest photosynthetic rate, concurrent with our previous knowledge of the higher density of the carbonizide dismutase photosynthetic enzyme, which makes the rate of photosynthesis greater than it is in shade-adapted plants. Finally, our results show that the lemonade berry had the highest stomata count, which did not agree with our previous findings. We suggest this might be caused by the fact that since stomata open in order to evaporate water, a plant grown in the sun might want to minimize this, since these plants are often also drought adapted and will want to maintain as much water as possible.
From the data collected, we believe that the tall evening primrose is both sun-adapted and shade-adapted. Primrose has low chlorophyll concentrations, low photosynthetic rate like the lemonade berry, low protein concentration, and low stomata count like the black sage plant.
The Tecolotta Canyon has been affected by global warming, from the warming of the environment from stronger sunlight to less water. As humans creating communities, building homes, and USD by the Tecolotta Canyon, there have been a bunch of impacts on the canyon's natural habitats, such as the loss of habitat and the threat to preservation of native species.
Thank you!